Hello students, in this presentation I will be discussing about the nucleus. We have already known that all living organisms are made up of cells. Cells are the structural and functional unit of an organism. These cells are of two types, that are the prokaryotic cell and the eukaryotic cell. The eukaryotic cell have a prominent structure which is called as the nucleus. So the word nucleus comes from a Latin word nucleolus, which means kernel or nut. So nucleus is a membrane bound organelle containing chromosomes and nucleolus. The nucleus is the brain of eukaryotic cell. Robert Brown in 1833 discovered a prominent body within the cell and termed it nucleus. Van Hamilton's experiment showed the role of nucleus in controlling the shape and features of the cell. Usually, the nucleus is round and is largest organelle in the cell. The nucleus varies in diameter from 11 to 22 micrometers. The nucleus typically occupies about 10% of eukaryotic cells volume, making it one of the cell's most prominent features. Generally, there is only one nucleus per cell, but nucleus is absent in the red blood cells of human beings and sieve cells of plants. Some organisms like paramecium and the tapetal cells of plant has two nucleus, and this condition is called as binucleate condition. Some organisms like slime molds, siphonales have more than two nucleus. This condition is called multinucleate condition. The multinucleate animal cells are called syncytial cells. Example, the epidermal cells of ascaris, and the multinucleate plant cells are called cenocytic cells. The shape of nucleus varies considerably. In most of the cells, it is spherical in shape. It may be cylindrical or elliptical. Now coming to the structure of the nucleus consists of four parts. The first is the nuclear membrane or karyotheca. The second is the nuclear sap or nucleoplasm. The third is the chromatin fibers, and fourth the nucleolus. First, the nuclear envelope. The nucleus is enclosed by a double membrane called the nuclear envelope or nuclear membrane or karyotheca. The nuclear envelope separates the nucleoplasm from the cytoplasm. Each nuclear membrane is about 75 to 90 Armstrong thick, and it is lipoproteinous in nature, means it is made up of the lipids and proteins. The two membranes remain separated by a space of about 100 to 150 Armstrong unit. As you can see here, this is the outer membrane of the nuclear envelope, and this is the inner membrane of the nuclear envelope, and these two membranes are separated by a space which is called as perinuclear space, and this space is about 100 to 150 Armstrong unit. The space between two membranes is called as the perinuclear space. The outer membrane is continuous with the rough endoplasmic reticulum, and it is studded with the ribosomes. The inner membrane is linked by fibrous filament proteins called lamins, which forms a scaffold of filaments called the nuclear lamina. The nuclear envelope is perforated with thousands of pores called the nuclear pore. As you can see here, the outer membrane and the inner membrane of the nuclear envelope, and in between these two membranes are perforated by thousands of pores which are called as the nuclear pore. The nuclear pore were first discovered by Callan and Tomlin in amphibian oocytes. The number of pores per unit area of nuclear envelope varies with cell type and with the physiological state of the cell. The nuclear pores are circular in view and have a diameter between 10 to 100 nanometer. The nuclear pore has a complex organization. So the entire structure of nuclear pore is called the nuclear pore complex. The nuclear pore complex consists of two rings or annuli with an inside diameter of 80 nanometer. A large particle present in the nuclear pore which forms the central plug. So the nuclear pore has a large particle in the center which are called as the central plug. 8 radial subunits or spokes extend from the inner and outer membrane where they join. Actually, they form a ring of subunits which are 15 to 20 nanometer in diameter. Each subunit project, so this is the central plug, and each subunit, this is the outer ring and this is the inner ring, and each subunit project a spoke like unit into the center. So the pore looks like a wheel with 8 spokes from the top. The nuclear pore forms an aqueous channel connecting the cytosol with the interior of nucleus. When materials are to be transported through the pore, it opens up to form a channel some 25 nanometer wide. So this channel is large enough to allow the passage of large assemblies like the ribosomal subunits. So nuclear pore complexes are active, that is they require energy for their function. All proteins synthesized in cytoplasm and those needed by nucleus must be imported into the nucleoplasm through the nuclear pore complex. So these substances are like the histones, ribosomal proteins, transcription factors and splicing factors all are imported into the nucleoplasm through this nuclear pore complexes. Similarly, molecules and macromolecular assemblies like ribosomal subunits, mRNA, tRNA are exported from the nucleus through this nuclear pore complex. The function of nuclear envelope is to regulate and facilitate the transport between the nucleus and the cytoplasm. Mainly it serves to separate the genetic component, that is chromosomes, from the protein synthesizing machinery, that is ribosomes and endoplasmic reticulum. So this provides protection to DNA against mutagenic effects of cytoplasmic enzymes. The nuclear envelope provides a surface for the attachment of structural elements of cytoplasm like microtubules and the filaments. The attachment of chromosomes to nuclear envelope assists in their condensation and separation during cell division. Nuclear envelope is involved in formation of endoplasmic reticulum and Golgi bodies. Now coming to the second part of the nucleus, that is the nucleoplasm. The nucleoplasm is filled with a homogeneous transparent acidophilic fluid called the nucleoplasm or nuclear sap or karyolymph. The nuclear component such as chromatin, heads, threads and nucleolus remain suspended in the nucleoplasm. The nucleoplasm is composed of nucleic acids, proteins and minerals. The most important nuclear enzymes are the DNA polymerase, RNA polymerase, NAD synthetase, alkaline phosphatase. In nucleoplasm, events like replication of DNA, transcription of RNA and transport of material occurs. Now coming to the nucleolus, the nucleolus means a small nucleus. So is a spherical ball-like structure found in the nucleus. It was discovered by Fontana. In cell biology, nucleolus is a suborganelle of the cell nucleus, which is an organelle. The size of the nucleolus is found to be related with the activity of the cell. Therefore, the cells which synthesize the proteins contains comparatively large size nucleoli. So, example are the oocytes, neurons and secretory cells. No membrane separates the nucleolus from the nucleoplasm. Calcium ions are supposed to maintain its intact organization. Nucleoli are made up of protein and ribosomal DNA or rDNA sequences of the chromosomes. The rDNA is a fundamental component since it serves as the template for transcription of ribosomal RNA or rRNA for inclusion into the new ribosomes. Nucleoli disappear during metaphase of cell division. After daughter cells complete separation, nucleoli reform around the nucleolus organizing regions of the chromosomes. During period between cell division, when chromosomes are in their extended state, one or more chromosomes have loops extending into the spherical mass called the nucleolus. The nucleolus is organized from the nuclear organizing region of different chromosomes. So you can see this is the chromosome and it is having a nuclear organizing region from which the nucleolus arises. The nucleoli are formed around the DNA loop that extends from the nuclear organizer. A number of chromosomes get together and transcribe RNA at this site. Here, three kinds of ribosomal RNA molecules, that is the RNA 28S, 18S and 5.8S are synthesized. These three types of rRNA molecules are used in the assembly of large and small subunits of ribosomes. 28S, 18S and 5.8S ribosomal RNA is transcribed by RNA polymerase 1 from the hundred of tandemly arranged rDNA genes which are distributed on different chromosomes. The rDNA containing regions of these chromosomes cluster together in the nucleolus. The nucleolus that is active in synthesis of ribosome typically exhibits three regions under electron microscope. The first is the fibrillar center. This pale staining part represents the innermost region of nucleolus. It is made up of a network of fine, about 4 to 5 nanometer thick fibrils. These fibrils are formed of rDNA, rRNA and ribonucleoprotein particles or RNPs. The second part of the nucleolus is the granular component. This is the outermost region of the nucleolus where processing and maturation of pre-ribosomal particles occur. The third part is the amorphous matrix. This is the proteinaceous ground substance in which the granules and fibrils remain suspended. So this is the structure of the nucleolus, which consists of the fibrillar center, the granular component and amorphous matrix. So nucleolus are mainly involved in the synthesis of the ribosomes. Next, coming to the chromatin fibers, the nucleoplasm contains thread-like, coiled and much elongated structure called the chromatin fibers. Chromatin fibers are observed only in interphase nucleus. During cell division, chromatin fibers become thick and ribbon-like structures called as the chromosomes. Chemically, chromatin is a nucleoprotein, that is, it is composed of the nucleic acid and the protein. Coming to the function of the nucleus. Nucleus is the dynamic center which controls and regulates various metabolic activities like growth, protein synthesis, metabolism and reproduction. The nucleus or the vehicle for transmission of hereditary characters through chromosomes during cell division. So nucleus plays an important role in the hereditary, that is the transfer of the characters from the parents to offspring. The synthesis of ribosomal RNA takes place inside the nucleus. So this is about the nucleus, which are membrane-bound organelle containing chromosomes and nucleolus, and the nucleus is made up of four parts. The first is the nuclear membrane or the karyotheca, and second the nuclear sap or the nucleoplasm, third the chromatin fibers, and the fourth the nucleolus. The nucleus controls and regulates different physiological functions of an organism.